Lecture 9 is all about how cells actually communicate the processes that they are trying to accomplish. We've talked about a number of these processes, the most recent of which was photosynthesis. So pause the video here and try the next 5 questions on Socrative. Come back here and I'll go through the answers. So what are the purpose of these light reactions of photosynthesis? Well, if you're going to be using light, recall that that light provides the energy that's necessary for these organisms to be able to actually photosynthesize. The electrons for this process are going to be provided by water. So we're going to be using the sun's energy and water's electrons to create ATP and NADPH, as long as we're doing linear electron flow. What are the purpose of the dark reaction, sometimes called the Calvin cycle, photosynthesis? Well, the first step of the cycle is to fix carbon. This is when the carbons for carbon dioxide are attached to larger organic molecules. In this case, the 5-carbon RuBP molecule that has 2 phosphates. And you'll recall that the enzyme that's responsible for this is known as Rubisco. It is absolutely true that plants have mitochondria. Even though this process of creating large organic molecules themselves using the energy from light and the electrons from water for photoautotrophs is an incredible process, you still have to be able to utilize the molecules that you've made, and mitochondria and cellular respiration are perfect for that. Now, how many turns of these dark reactions of that cycle is necessary to produce 1 glucose? Well, recall that when we did glycolysis, you took a 6-carbon molecule and at one point broke that 6-carbon molecule down into 2 3-carbon molecules known as G3P. The yield for 1 turn of the Calvin cycle is 1 G3P. So there's going to be 2 turns of these dark cycles in order to create a single molecule of glucose. And if you have to do 2 turns, each one requiring 9 ATP and 6 NADPH, that means 18 ATP and 12 NADPH are necessary to create a single molecule of glucose. If you were to go all the way back and think about the products of glucose when broken down in the mitochondria by aerobic cellular respiration, you might recall that there are going to be 10 molecules of NADH, an electron carrier, just like NADPH, 2 molecules of FADH2, and 4 molecules of ATP made by substrate level phosphorylation. There's a significant amount of energy here, which is lost in this process, because even though we can create and hold all of this energy, all this free energy in a large molecule of glucose, some of it gets lost to entropy during that process of breakdown. Now today we're going to talk about the sources of chemical events of these communications, both the signals and the receptors that receive them, how those signals go from their actual receival to a place where they can achieve a change inside of the cell, and then how we can regulate that process. You might recall that cells have actually gone through a process in which they've received signals in a way we've kind of seen before across membranes. So I want you to pause this video here and actually take a look at this video from TED Ed. It might surprise you to learn that the reason your muscles get tired is because their signaling events simply wear out. The ions that are responsible for cellular communication get used up and can't replenish fast enough in cells that aren't used to that process. When we talk about this process of communication, we're going to be using some very specific terminology. The signaling molecules are known as ligands, and the proteins that they bind are known as receptors. It's most common that these receptors are bound into membranes, and so they are part of the functionality of these membranes. And the most important ones that we're going to be talking about today are going to be receptors within that plasma membrane. These allow the cell to receive a signal from the outside and result in a change inside. So when you think about these chemical signals, these proteins that need to exit the cell to talk to neighbors, which of these processes accurately describes how chemicals, proteins produced inside the cell can get to the outside. Well, phago means to eat. So phagocytosis would be the eating, which is an intake of large molecules. Endo also means internal. So this is a process of making things internal. And transport vesicles are responsible for moving things within the cell. Exocytosis is the process by which we take internal products and allow them to exit. So which of these would be true regarding protein ligands that are secreted from the cell? And pause the video here if you need some time to think about it. Well, if you recognize the process of exocytosis as being very similar to the endomembrane trafficking system, you might also recall that proteins which traffic through that endomembrane system to the plasma membrane are glycoproteins. So we would say that these are glycosylated. And which of the following best describes the receptor that receives a chemical signal on the cell surface? Yes, it's probably going to be glycosylated, but let's think about that protein and how we can better describe it. Well, in order to be able to communicate what's happening on the outside to the inside of the cell, you actually need to be integrated into the membrane. So we would call this an integral, or sometimes known as transmembrane protein. Now, cells can actually receive signals that are going to be directly communicated by their neighbors. And so this is known as direct signaling. When the neighbor is secreting the signal into the local environment, this is known as paracrine signaling. When the cell signals to itself, this is known as autocrine signaling, where auto means self. Some secreted signals would be steroids, a class of lipids. So which one of these organelles would be responsible for the synthesis of a lipid? Hopefully you recall that this is the smooth ER. That's why cells like your liver, for example, have lots of smooth ER. Well, when these hormones, these lipid molecules, are produced in multicellular organisms like plants and animals, these hormones will travel through that multicellular system through the endocrine system. And so we call this process by which these signals are secreted in one part of the organism, but act in cells at another part of the organism, endocrine signaling. It's also very important to note that when you have an organism, a multicellular organism that can do endocrine signaling, not every cell in the body will actually express the receptors for that signal. For example, when you're undergoing a growth spurt, you don't necessarily want every part of your body to grow. You want the growth plates in the parts of your bone to grow. And so only those cells will express receptors for those hormones. Now we're going to take a look at 3 stages for cell signaling. It does not matter what type of cell or what type of signaling, the same processes occur for those signals. First is reception. The signaling molecule, the ligand, binds to the receptor. And there's going to be some way in which that receptor changes in response to that binding event. Remember that shape equals function. So if something is happening, a shape needs to change. Transduction is going to be when the shape change in the receptor causes a change internally, and it's going to carry that signal to another part of the cell. This signal transduction pathway leads to an area where there's a response which can occur, and that transduced signal results in a very specific response. Transmembrane receptors, so embedded into the membrane of the cell, these integral proteins, these are going to be of 3 types. The first one that we're going to talk about is a G protein coupled receptor. And just exactly like it sounds, it's a receptor that acts coupled to or paired with a G protein. G proteins are named because they use GTP. So recall that GTP is a product of the citric acid cycle, although we count it as an ATP in this class. But this GTP, just like ATP, is capable of providing free energy to a molecule, and that addition of energy can cause a shape change. So the ligand is going to bind to the G protein coupled receptor, causing a change in the receptor shape, which recruits a GTP and that G protein, and that GTP gets replaced on the protein, allowing it to become active. These are incredibly abundant in human cells, and about 60% of medicines that you take on a regular basis actually act on these G protein coupled receptors. In addition, diseases like cholera and botulism, these are toxins that actually interfere with the activity of G protein coupled receptors. Receptor tyrosine kinases are kinases that can act as receptors. These guys are going to do something a little bit different. Instead of having a special protein that they act with, two receptors will receive two ligands and then dimerize. Remember that di means two. So these are two of the same receptor tyrosine kinases coming together. These are kinases, which means they phosphorylate things, and they're actually going to phosphorylate each other. And then the phosphates on these activated dimers are going to be capable of starting multiple cellular responses because we have multiple phosphates that can be used here for this process of phosphorylation. The last one that I want to show you is a ligand-gated ion channel. This is actually something you've seen before. This is something that can be a role in active transport or just in controlled passive transport. A ligand-gated ion channel is going to be a channel that is opened only in the presence of the appropriate signaling molecule. One final one that we're not going to give any examples of, but I just want to make you aware. All three of the types of receptors I've shown you are embedded in the outer plasma membrane or into one of the internal membranes, depending on the signal response. There are intracellular receptor proteins, and these intracellular receptor proteins are oftentimes responsible for the receiving of signals such as hormones when these hormones can pass through the plasma membrane. These intracellular receptors don't have a transmembrane domain, they're not anchored in a membrane, and they're only going to be able to receive very specific types of signals because their signaling molecule has to be able to cross the membrane. Once that signaling molecule finds its receptor, now we have to transduce that signal because the receptor is located in one part of the cell, but the response is going to be located in another. Protein kinases are very important in this process because kinases are capable of phosphorylating other compounds. When you phosphorylate another kinase, you can create what's known as a phosphorylation cascade, like the picture that you see here. This is when a kinase becomes phosphorylated, creating an active version of that kinase. Now that kinase can phosphorylate another kinase, creating an active, which can phosphorylate another kinase, creating an active kinase. It's a series known as a phosphorylation cascade. But this is also an on-off switch. Phosphorylation can turn molecules on, but dephosphorylation can reverse this cascade and turn a molecule off. Enzymes known as phosphatases remove the phosphates from proteins in this reversal process known as dephosphorylation. This name phosphatase can be confusing. Recall that you already know kinases like phosphofructokinase and glycolysis, and you know that it transfers phosphates. Protein phosphatases are responsible only for the removal of phosphates. Now, there are going to be other types of molecules that can participate in this process, and if you're not a protein, then we call you a second messenger. So these are going to be molecules that are capable of interacting and participating in transduction, but they're not going to act as enzymes like kinases or phosphatases. Now, adenylyl cyclase is an example of an enzyme capable of creating one of these second messengers. It actually converts ATP into a small molecule known as cyclic AMP or CAMP. This is an example of a very common second messenger, and you're going to see an example of that in some of the processes we are going to look at. So here's an example of a signaling cascade. So which type of transmembrane receptor is this? Good. This would be a G protein coupled receptor because it's interacting with a G protein. That G protein gets activated, and you know that it's activated here because it has a GTP bound to it. That G protein, which is active, is going to activate adenylyl cyclase. That enzyme is responsible for the conversion of ATP into a non-protein second messenger known as cyclic AMP. That cyclic AMP can activate a protein kinase. Well, kinases transfer phosphates. So if kinase A phosphorylates another kinase, we call this a phosphorylation cascade. And eventually that cascade of different kinases can get all the way down to a cellular response. A cell can regulate this process pretty easily. For example, if you inhibit adenylyl cyclase right from the get-go, everything that happens after that would already be tightly controlled. Here's an example of one of these cell cycle pathways that can occur in disease. Vibrio cholera is a bacterium which causes the disease cholera by producing a toxin. The cholera toxin binds to a G protein and modifies it so that that G protein gets stuck in the active conformation, meaning that GTP is bound to that G protein. Since that G protein activates adenylyl cyclase, it causes an excessive production of cyclic AMP, activating the gated ligand or the gated ion channel, and so we're going to be pumping chlorine ions out of the cell. If cholera is found in your intestines, this results in an excessive amount of these ions being secreted out of your intestinal wall. When you have an excess amount of these solutes in your intestinal cavity, this also causes the water from your cells to osmoregulate out of the cells. And so you get this watery diarrhea, which is what's caused by cholera. And so that's what the cause of this type of disease is. Now, ions are not just crossing your plasma membrane. They can actually be another example of second messengers. Calcium, which you might recall from watching the muscle video that I introduced you to, is another common second messenger. And one of the cool things about calcium is that, unlike the example that you just saw with cholera, calcium can be secreted because it's stored in the smooth ER in high concentrations. And when it's released, the entire cell gets flooded with calcium ions. And so it can actually result in multiple signaling pathways being activated at once. And it actually allows the entire cell to be regulated. For example, when multiple nerve cells talk to one another. Go ahead and review this video to see this process for yourself before coming back to view the second video in this lecture series.